In this video, I'm going to discuss pretty much everything you need to know about the Kreb cycle, otherwise known as the TCA cycle or the citric acid cycle. I'm going to call it the TCA cycle from now on, since that's what I prefer. And TCA stands for tricarboxylic acid. So the tricarboxylic acid cycle. Let's get started. So as we know, from glycolysis, glucose is converted to pyruvate. And remember, there's two pyruvates. So from this point on, everything is going to happen twice because there's two pyruvates that are produced by glycolysis. So pyruvate is transported through the outer mitochondrial membrane through a pyruvate symporter, which brings in a H plus ion as well. So just keep that in mind. And so pyruvate is now inside the inner mitochondrial membrane space, and the inner mitochondrial membrane space is the location of the TCA cycle. So pyruvate gets converted to acetyl-CoA. That's the first step of the TCA cycle. And just know that this is a one-way reaction, and this will help you when you're thinking about, why can't we just go back from acetyl-CoA to pyruvate and then go into gluconeogenesis? That's not possible because this is a one-way reaction. So that's something that confused me. So hopefully that clarifies it for you. So we've got pyruvate to acetyl-CoA, and in this process, an NAD is converted to an NADH. And because pyruvate is a 3-carbon molecule, but acetyl-CoA is a 2-carbon molecule, we have to lose a carbon. So we lose the carbon dioxide here. And the enzyme that performs this reaction is the pyruvate dehydrogenase complex. So let's look at how this pyruvate dehydrogenase complex is regulated. So enzyme 1 is regulated by increased levels of ATP. So because it's a complex, there's actually 3 enzymes inside it. So that's why we're going through 3 different enzymes. So as I said, enzyme 1, high levels of ATP will inhibit the pyruvate dehydrogenase complex. For enzyme 2 of the complex, increased levels of acetyl-CoA and increased CoA. So increased acetyl-CoA will inhibit, but increased CoA will promote the reaction to go forward and will stimulate enzyme 2, while enzyme 3 is inhibited by NADH, but is stimulated by NAD. And this makes logical sense because if you have a lot of acetyl-CoA, you're not going to need this reaction to go forward because you already have a lot of the product. And the same with this reaction here. You already have a lot of NADH. So if you have a lot of it, then the reaction doesn't need to go forward because we already have a lot of the product. So that's how I kind of remember the regulation there. And even if the understanding part isn't fully concrete for you, I would recommend just going through and memorizing those 3 enzymes, writing it out a few times until it sticks. And then hopefully the more that you write it out, the more sense it'll make in the context of the whole cycle. So speaking of the whole cycle, let's continue. So we have acetyl-CoA, which is a 2-carbon molecule, which then becomes just a CoA, so this gets converted. This is just how the first step goes, and simultaneously an oxaloacetate is converted to a citrate, and also a fluoroacetate becomes a fluoroacetyl-CoA and then becomes a fluorocitrate. So there's a few reactions going on. There's 3 going on. So from fluoroacetate to fluoroacetyl-CoA, we need a CoA there, and then that becomes fluorocitrate. And fluorocitrate, we'll see, is important in a second. So when we look at oxaloacetate to citrate, that enzyme is citrate synthase that is used there, and we have to add a water here because that's what the reaction requires. And this is inhibited by NADH and succinylcholine, which is another substrate of the TCA cycle. I'm not too sure why, but that's just how it goes. So I would just commit that to memory because regulation is always really important. So as I said, fluorocitrate is about to become important. Fluorocitrate is quite toxic actually, and in the next reaction between citrate and isocitrate, which is catalyzed by aconitase, fluorocitrate will inhibit this reaction. So that's why I wrote it up here. Even if you don't understand this full process, I would just remember that aconitase is inhibited by fluorocitrate. So the next reaction is between isocitrate and alpha-ketoglutarate. in this reaction, an NAD becomes an NADH, and the enzyme here is isocitrate dehydrogenase. Now, isocitrate is a 6-carbon molecule, and that means that in this process, we're going to lose a carbon dioxide because isotrace has 6 carbons while alpha-ketoglutarate has 5 carbons. So we're going to lose a carbon here through the carbon dioxide. So isocitrate dehydrogenase is a regulation of this. It's promoted by calcium ions and ADP, but it's inhibited by ATP and NADH. So I write that out a few times until it sticks. And let's move on to the next reaction. So now we have alpha-ketoglutarate to succinyl-CoA. And this is facilitated by the enzyme alpha-ketoglutarate dehydrogenase complex. So it's similar to the complex we saw up here. It's made up of a few enzymes, but we're just going to consider it as one big complex for simplicity's sake. So in this process, because it's a dehydrogenase, we've got an NAD to an NADH. We're going to lose another CO2 because we're going from a 5-carbon alpha-ketoglutarate to a 4-carbon succinyl-CoA. So I'll just put that here to lose that carbon dioxide. And then we're going to add a CoA in this process because the succinyl-CoA has a CoA in it. Now the regulation of the alpha-ketoglutarate dehydrogenase complex, calcium ions promote this enzyme, while NADH, ATP, and succinyl-CoA inhibit this enzyme. So I recommend you commit those to memory again. Then next up we have succinyl-CoA to succinate. So this requires energy. So the enzyme here is succinate thiokinase, or you may see it written as succinyl-CoA synthetase. In this case, I think it's worth memorizing both because either one could come up in an exam. So we have a GDP plus a phosphate being converted to a GTP, and we have to remove the CoA that we put in to get to succinyl-CoA. So we've got to remove that back out here because succinate doesn't have a CoA. So this requires energy, this process, as I said, so it's important. Then we'll move on to the next step, which is succinate to fumarate. Now, this is facilitated by the enzyme succinate dehydrogenase. And succinate dehydrogenase is really important because it is one of the complexes of the electron transport chain. To be more specific, it is complex 2. And in complex 2, if you look at the electron transport chain video, an FADH becomes an FAD, but in this case, an FAD is becoming an FADH. That's what its function is in this cycle. So we're producing an FADH, and myelinate is a competitive inhibitor of succinate dehydrogenase. So make sure you commit that one to memory. Then from fumarate to myelate, we have the enzyme fumarase. And in this reaction, water is added. And just keep in mind that from now on, all these substrates are 4 carbons. That's the length of their carbon chain. So then finally we have myelate to oxaloacetate, and this enzyme is myelate dehydrogenase. So we've got an NAD to an NADH, and this is inhibited by NADH, this enzyme. So that is the TCA cycle and how it's regulated and the important points. But just before we finish up, let's look at how many ATP are produced from the TCA cycle. So let's recall. So FADH are worth 1.5 ATP. That's just what one source says, and that's what I was told to remember from my lecturer. And an NADH is 2.5 ATP. So if we consider this, let's count up how many NADHs we have. So we've got to multiply everything by 2, remember, because if we look at up here, we've got 2 pyruvates from glycolysis. So we've got to go through the cycle twice. So let's count up how many NADHs we have. So we've got 1, 2, 3 NADHs. Multiplied by 2 is 6. So we have 6 NADHs. Let's see how many FADHs we have. We've got 1, but we have to multiply that by 2 because we're going through the cycle twice. So we have 2 FADHs, and then we have 2 GTPs. So we've got 1 here that's seen in the cycle, but we're going through twice because there's 2 pyruvates at the start, and therefore we have 2 GTPs. Then to find the total ATP that is produced from the TCA cycle, we go 1.5 ATP times the 2 FADHs, 6 NADHs times 2.5 ATPs, plus 2 GTPs, which are pretty much equal to 1 ATP. And the total that you get is 20 ATPs when you calculate that through. And just consider that that means that 1 acetyl-CoA means 10 ATP, but 2 means 20 ATP. So for this cycle, it is 20 ATP. And the reason why I asked you to consider that 1 was 10 ATP is because later we'll see that that will help us when we're calculating ATP for beta oxidation. So that is the Krebs cycle regulation, all the enzymes, and the total amount of ATP. Let's just have a quick look now at entry and exit points to the TCA cycle, just so we can truly understand where this sits when we think about it in relation to other cycles. So the entry points to the TCA cycle. Acetyl-CoA can come from glycolysis. Oxaloacetate and malate can come from anaplerotic reactions. I have another video on that. So anaplerotic reactions basically convert the end products of glycolysis and glycolysis to oxaloacetate and malate. So we've got the end products of glycolysis, which is pyruvate and PEP, and it will convert those to oxaloacetate and malate, which feeds into the TCA cycle. Succinylcholine can come from odd-numbered fatty acid breakdown, so I have a video on that, or from the use of ketone bodies. So I also have a video on ketone bodies. I don't really show in that video where the succinylcholine comes in, but just know that ketone bodies can be converted to succinylcholine and enter the TCA cycle. Also, alpha-ketoglutarate can come from the deamination of amino acids, and we'll talk about that in a bit more detail when we look at the urea cycle. Finally, just some exit points from the TCA cycle. We have citrate, which leaves and goes into fatty acid synthesis, which I'll have another video on. Alpha-ketoglutarate, which can go into amino acid synthesis, which you'll see in the urea cycle. Succinyl-CoA can go into heme synthesis, which I don't have a video on because I didn't go through that in my course, but just know that it can go to make heme in the body, which is really important. And then we have malate and oxaloacetate, which can exit and go into gluconeogenesis. So those were the entry and exit points to the TCA cycle. And then now I just want to quickly go through how I remember all of this, because it can be quite overwhelming at first when you've got that big diagram. So how do I remember the substrates? Here's my mnemonic. Can I keep selling substrates for money, officer? And in an exam, I usually just write the first letters to trigger my memory. For the enzymes, my mnemonic is Andy did dance, so Dave Franco did cycling. Now this is going to need a bit of explanation, so I start at aconitase, and then all the D's stand for dehydrogenases, and all the D for dehydrogenases involve an NAD becoming an NADH, except for Dave, because Dave is different, and that represents succinate dehydrogenase having an FAD becoming an FADH. So that's how I remember the enzymes, but I just start at aconitase because that's easiest for my brain. You can make up your own mnemonic if you'd like. So that's, and I just write the letters as usual. So I also have a mnemonic for the regulation of the TCA cycle, and that's please Andy, I know Sandy more closely. And I just write the letters again. So P stands for pyruvate dehydrogenase complex, A stands for aconitase, I stands for isocitrate dehydrogenase, K stands for alpha-ketoglutarate dehydrogenase complex, S stands for succinate dehydrogenase, M stands for malate dehydrogenase, and C stands for citrate synthase. And once I know which enzymes are regulated, which is what this mnemonic helps me out with, then I can fill in what regulates them. And that just helps me personally to remember them. But I definitely recommend mnemonics to help you out. So that was everything you need to know pretty much about the TCA cycle.